Hello, everyone. Welcome to the African American chapter event brought to you by Myeloma Community by HealthTree program. My name is Valerie, and I'm the African American chapter lead. Now, it may seem like I'm hiding from you guys today, but I'm not. I just had to step away to where I don't have a very good internet connection. And I find that when I keep the camera off, it comes through a little bit clearer. So I am here with you in bright spirits, and I'm glad to see each of you that's able to join us today. So thanks. Now, today's session is going to be recorded. The recording along with the slides are going to be sent to all registrants within 48 hours after the event has concluded. Now, as always, I'd like to thank our sponsors, without whom this would not have been possible. Bristol Myers Squibb, Takeda Oncology, Karyopharm Therapeutics, Adaptive Biotechnologies, Oncopeptides, Janssen Oncology, AbbVie, Sanofi Genzyme, Amgen Oncology, and Genentech. The agenda for today is as follows. We're going to hear from Dr. Scott Goldsmith, who I'll introduce formally in just a moment. After Dr. Goldsmith's presentation, we're going to open the meeting up to a Q&A with the audience. And as part of our outro announcements, Anna Sahagun from the HealthTree Cure Hub team will share with us a little bit about the genomics study. Now, today's topics will be Genetics 101 with multiple myeloma specialist, Dr. Scott Goldsmith. Knowing your genetic profile is crucial to getting the best care. This knowledge will contribute to knowing your risk profile, finding the most appropriate treatments, and determining the length of recovery. Dr. Goldsmith has generally offered to speak to us in a simple way about a complicated topic so that we can better understand our disease and empower ourselves to receive the best care. It's now my pleasure to introduce to you our featured speaker. Scott Goldsmith specializes in the treatment of multiple myeloma, which is also the focus of his prolific research. Dr. Goldsmith has been quoted as saying, "I am passionate about bringing new and innovative treatments to patients with multiple myeloma and other hematologic malignancies through translational research and clinical trials." Dr. Goldsmith entered medicine because he remembers how well he was treated when he was a patient in his teen years. Dr. Goldsmith says he felt firsthand the profound, lasting impact that healthcare professionals can have on an individual. A Florida native, Dr. Goldsmith graduated with honors from the University of Florida, Gainesville, then earned his medical degree from UF's College of Medicine and continued his training at Barnes-Jewish Hospital, Washington University School of Medicine, St. Louis, Missouri. Dr. Goldsmith takes pride in the relationship he forms with his patients, and he is intent on making an impact at an individual, institutional, and global level, finding better treatments for multiple myeloma. So what we're going to go into is the presentation with Dr. Goldsmith. So Dr. Goldsmith, the floor is all yours.
Okay, I'll go ahead and share my screen. And please let me know. You can see it, I assume. Great.
Yes.
So thank you, Valerie, for the kind introduction. I'd really like to extend my sincere gratitude to Audrey and Greg Brosite with Myeloma Crowd, as well as Myeloma Crowd itself, and this chapter for allowing me to talk about a couple of different subjects that I'm actually really interested in and passionate about. Today, we're going to try to cram a couple of college courses into a 30-minute session with some Q&A. but we'll try to keep it a pretty high level so that it can be understandable at any educational level, any audience, because I think it's really important that everybody has a foundational understanding of where medicine is going and how it relates to their individual treatments, not only for multiple myeloma, but any disease. So we'll discuss a little bit of the basics of genetics and genomics, then talk about how it relates to patients with multiple myeloma and the African American community. I really encourage everybody to ask questions either during or after. And if I can't get you the answer now, I promise I will get you the answer later. So with that, I do want to give a little bit of an outline for what we're going to talk about. We'll start by presenting a case that I think provides a really nice framework that encompasses all of these different topics into in this presentation. We'll then go into a genetics overview, understanding the different sciences that are that study genes and what the definitions of everything are. We'll talk about genetic mutations and how that relates to disease, and then how we can target them in a lot of different branches of medicine. And then we'll focus in on multiple myeloma and how it relates to the treatment of multiple myeloma patients, and make sure throughout all of this that we're keeping relevance to the African American community and African American patients. We'll conclude with a question and answer session. And like I said, if you do have questions that are pertinent to a specific slide, one of the moderators can interrupt me at any time. So we'll start with the case. This is a patient that I took care of during fellowship and kind of stuck with me because, like I said, it really encompasses all of these interesting aspects of this topic. So she was a 62-year-old Black woman with multiple myeloma who had achieved a complete remission after induction chemotherapy and autologous stem cell transplant. But she had what we call an early relapse, only 18 months after her transplant, while still on maintenance therapy. And unfortunately, she didn't just have biochemical relapse, she had new lesions in her bones. So this was obviously concerning. So she came to us to discuss options. And so this was when I was at Washington University. We discussed options that were both standard of care, but also clinical trials that were available to her. And one that stood out to her was this study called the MyDrug study. Essentially, it was a clinical trial where we were trying to personalize the therapy specifically to any genetic drivers that she might have. And this stood out to her, so she decided to enroll in that clinical trial. We looked at her bone marrow. We took a bone marrow biopsy and then sent it off for what we call next generation sequencing, where they can sequence all of the genes in the myeloma cells, especially the ones that are involved in disease. And this is to look for genetic drivers that we might be able to target. We could target these potentially with drugs that are already on the market, potentially for other cancers. And fortunately, we found one of these driving mutations. This was called BRAF V600E. And this is a mutation that's found in many cancers. We know it's found in multiple myeloma, but nothing is specifically used in multiple myeloma for that mutation. It's more for melanoma, the skin cancer, also colon cancer. But as part of this clinical trial, we were able to use this melanoma drug called cobimetinib as part of her treatment. And she started on that drug alone and achieved a response within the first cycle, which was really encouraging to us and kind of a proof of principle that we can target myeloma mutations with drugs that we already have. And what's on top of that is that we were able to combine myeloma therapies in subsequent cycles, and they had synergy. They combined together, and she achieved this really good complete remission that lasted for a long time. So this is one area of myeloma therapy that seems to be the future of, or one direction that we're heading in, which is personalized, tailored therapy based on the genetics of their disease. So that's why it's so important for all of us to have a better understanding of what genetics and genomics are, and how what direction the medical and myeloma community is moving because of that. So let's start with just some foundation of genetics. And really the place to start is with the definition of a gene. And it's not quite as simple as one might think. There are a couple of different definitions of a gene, and this is really the foundation for numerous branches of science, but they all interrelate. So it's important to understand both. So one definition of a gene is the sequence of DNA, the blueprints for life that carries the instructions for making functional proteins or functional molecules in the body. Another definition of a gene is a unit of heredity. It's something that's carried on from parent to offspring, and it determines a specific trait, or in combination with other genes, a bunch of traits, or can contribute to genetic diseases. And so these are different definitions, but they interrelate. And so it's important to understand both. And I think we'll start by diving into the molecular biological aspect or definition of a gene, and that's how it relates to DNA. So let's just talk a little bit about what DNA is and what DNA does. Some people refer to DNA as the building block of life, but I think of it more as a blueprint. DNA is this double helix molecule found in every cell, and it contains the instructions for essentially each cell to make functional proteins and allows the body to function. And so DNA is this code that's made of kind of variations in 4 different letters, or what we call bases, just different repeating sequences called that are C, G, A, and T. And so these contain the code or the instructions for your cells to make functional molecules. And so there are 2 main processes that happen with DNA. The first is the DNA can be replicated, it can be copied, and this allows cells to divide. The other thing that can be done is a message can be transcribed from these instructions. The message is called RNA. RNA is a single strand using 4 different letters, and this message is kind of an intermediate. The message leaves a certain part of the cell where it then can be translated by the machinery of the cell. It's translated into usually proteins. These are the functional molecules that do they're the business end, the workhorse of each of the cells. It can be enzymes or structural proteins. It can code for a bunch of different things. And this is where, this is, like I said, the business end of things happening in the cell. So this is what we call the central dogma of genetics or molecular biology, is we take the DNA, which is the code, we transcribe it into a message and then move it to a different location into the cell. And then the machinery of the cell uses that message to produce proteins, which are kind of the building or kind of the workhorse. So now that we understand what DNA is and what DNA does, we can kind of move into the definition of a gene, and then also how the DNA and the genes are packaged. So a gene in the biological sense is a region of the DNA that codes for some of the proteins. And so it's a string of DNA molecules in a row. So just as some interesting facts, all of the DNA in one cell, if you were to string it out all along, would actually be longer or taller than a person. If we were to take all of the DNA that's in every cell and then lay it out flat, it would actually go for 67 billion miles. That's how much DNA is contained within one human. And so that's equal to 150,000 round trips to the moon. So obviously you have to wonder, how can that all be packaged into each individual or each cell? There's just so much of it. And this is where we get into the other definition of genetics and genes, and we'll go into that in a little bit. But the genes are packaged in these packages called chromosomes. They're wrapped tightly around some proteins and then organized in these structures called chromosomes. The chromosomes are contained within the nucleus of the cell. So all of the DNA in your cell, for the most part, is contained within the nucleus in these structures called chromosomes. So if we're talking about packaging, we also have to talk about how these packages are shipped. And they're really shipped from parent to offspring. We have 23 pairs of chromosomes. So that's where you might hear the 23andMe. One set of chromosomes comes from a father, and one set of chromosomes comes from a mother. And so this is how genes are packaged in different locations. And that implies how sometimes those genes are inherited. They come in packages with other genes on chromosomes. And so that gets important, has important implications for genetic or hereditary diseases. You have 20 to 30,000 different genes throughout the body, but that makes up like maybe 1% of all of the DNA within a single cell. A lot of it doesn't code for anything. The majority of it just kind of has unknown functions or functions that don't lead to proteins. So the genes are the ones that do code for proteins, and all of these genes make up one's genome. So when we talk about a genome, it's all of the genes that are involved in making you. So that gives us a little bit of a overview of what a gene is from two different definitions. One, the gene that is a sequence of DNA. Secondly, a gene that is just a package inherited from parent to offspring. And so these definitions are kind of the foundation for two different branches of science, once again, that have some overlap, but are studied somewhat differently. This one, the study of heredity, is genetics, where you study genes and how they are inherited and how traits or diseases are passed down. The other study is genomics, which is the study of all the genes in an individual and how they interact with each other and how they interact with a person's environment. And so there are some overlap, but they carry some distinction as well. So with a little bit of a foundation of understanding genes, genetics, and genomics, now we can understand a little bit about how things go wrong and where they go wrong, when mutations occur, and then what we can do about it from a medical standpoint. So mutations can occur at a small level or at a larger kind of chromosomal level. So we'll start here, where there can be a change of just a single letter or a single base in the sequence of your genes. It can be a substitution where a gene changes to something that it shouldn't be, or a letter changes to something that it shouldn't be. A letter can be inserted into an area where it shouldn't be, or a letter can be deleted. And these obviously have downstream implications, because if you think of a word and you change the letters that are in it, that meaning can change dramatically. So the message that you get and then the protein that you get from this blueprint, from this code, can change dramatically. And that can lead to disease or dysfunction in the cells, all with just the change of one of these bases, one of these letters. But in addition to these small single point mutations, you can have larger chromosomal events, chromosomal mutations. We call these cytogenetic abnormalities. And they can occur in a number of different ways. You can have large chunks of chromosomes deleted. You can have them flipped around or inverted. You can have them translocated, where a part of a chromosome from one chromosome is put on another place. And as you can imagine, that can have a lot of downstream effects as well. Specifically with translocations, what can happen is, and this happens a lot in multiple myeloma, you can have a gene that's on one chromosome put near what we call a promoter, something that increases the expression of that gene. And then that can have downstream effects and lead to malignancy or other issues. And so mutations can occur at a very small level with just one letter, or they can happen at a larger level with big chromosomal events. And so that's kind of the key point I want you to take away from this slide. So let's talk about how these mutations can occur. At what time point in one's life can these mutations occur? So when we think of genetic diseases, diseases that are inherited from one person to another, we call these mutations germline. They can be inherited because a parent is a carrier of that mutation, or in the what we call the germ cells, the sperm and egg, the mutations can occur at that time, and then at conception that all of the subsequent cells would carry that mutation. So we consider these kind of the genetic mutations that contain these germline events, and the subsequent disease is related directly to those mutations. So one disease that is unfortunately all too prevalent in the African American community and very relevant is that of sickle cell disease. And it is a good example of how a genetic disease is passed down. So an individual may be a carrier of the sickle cell trait because they contain a mutated hemoglobin gene on one copy of their chromosome. They have another chromosome that contains a normal copy. So likely throughout their life, they would be asymptomatic or minimally symptomatic because they have enough functional hemoglobin. If they find a partner who is also a carrier of the sickle cell trait or any type of genetic trait, and then have children, those children, each child is at a 25% chance of getting 2 normal copies, a 50% chance of inheriting a normal copy and a mutated copy, and unfortunately a 25% chance of inheriting 2 mutated copies. Those individuals that get 2 mutated copies in this particular mutation are pretty much unfortunately guaranteed to suffer from the disease of sickle cell, because what this is due to at an underlying DNA level is a single letter substitution that leads to a downstream change in how the protein structure is. And then the protein being hemoglobin, which is the carrier of oxygen in all of the red blood cells. When there's that mutation, these proteins then start to aggregate and cause the cells to sickle. They sickle, and as they're passing through the bloodstream, they tend to combine with one another and cause vasoocclusion or blockage of small blood vessels. And they also have the predisposition to hemolyze or break up, and that leads to the anemia aspect. So an individual who carries 2 copies of this mutated hemoglobin gene, unfortunately, is almost 100% likely to have the disease itself. What's the one good thing about the understanding of this disease, both at a genetic and a molecular biology level, is that we're starting to understand how we can go about reversing this. And there are a number of therapies in studies at the moment where they're aiming to change this gene back to the normal one in a patient's own stem cells and then giving those stem cells back, or changing other genes, flipping other genes on or off that allow for a different type of hemoglobin to be more prevalent and prevent all of these downstream effects. So we're really early in that sort of science, and obviously there really needs to be rigorous oversight in the conduct of these trials and the development of these treatments. But it is somewhat exciting how we go from understanding a disease but not really able to doing anything about it to now we're starting to get the tools to do something about it. So that is an example of a genetic or an inherited disease in kind of the classical sense. So cancers, by far and away, aren't inherited specifically. However, an individual can inherit what's called a cancer predisposition syndrome, where they inherit one gene that makes them much more likely to develop another mutation that would then make them likely to develop cancer. So one example of this is the BRCA mutations. In this particular instance, a patient or an individual would inherit a mutated gene in the BRCA gene. This BRCA protein is involved in repairing DNA. So if it's involved in repairing DNA and you only have 50% of it actually working, then you're more likely to get more damage to your DNA going forward. It's not going to be in every cell, but it's going to be in some cells, cells that tend to divide or age more easily. And when you get that second damage, that leads to a cascade effect where you're much more likely to develop a malignancy from that. So you're inheriting this predisposition in your from your germline with one damaged gene, and then you acquire what's called a somatic mutation somewhere in your body. So we call this the second hit. And unfortunately, these patients are at much higher likelihood of developing breast cancer at an early age, and as they get older, a much, much higher incidence of breast cancer, as well as ovarian cancer, prostate cancer, and pancreatic cancer. But it's not a guarantee that they're going to develop this, and this is because they inherited this predisposition. But unlike sickle cell, in which all individuals have two mutated copies that directly leads to that disease, they need to acquire a second mutation. And so that's why they're at risk of it, but not necessarily guaranteed to have it. So because we know about what this is obviously a very busy slide and maybe a little bit out of the scope of this talk, I include it mainly as just an example of how our understanding of the genetics of the disease and then the downstream effects allow us to maybe target it. We know that the BRCA mutation is responsible for DNA damage. Normal cells have at least one copy of the normal BRCA repair mechanism. So we can give patients an inhibitor of a backup mechanism, and their normal cells should be able to handle that. Whereas the cancer cells, which have a complete loss of this DNA repair machinery, if you take out all of their backup mechanisms, then that leads that cancer cell to die. And so, like I said, because we have a better understanding of the genetics and then the downstream effects, we can design more tailored treatments specifically for patients with those mutations. So I talked about genetic diseases that are inherited. I talked about cancer predisposition syndromes, where you at least have to acquire, or you at least have to inherit some aspect of it. But the vast majority of cancers and diseases are due to acquiring multiple somatic mutations, multiple acquired mutations throughout one's life at some point, as well as other genetic events or exposures. And so we're starting to uncover what those genetic drivers are, but it's going to be different from patient to patient. And as I said, the majority are going to be acquired throughout one's life. So this is where multiple myeloma comes in. We do know that there is some familial predisposition in some groups, including African Americans, who are more likely to have a relative who has had multiple myeloma in the past. We haven't yet uncovered what it is that they're passing down or what they're inheriting, but it requires more than just that. It requires a number of other mutations down the line. Plasma cells, which are the normal version of the cancerous myeloma cell, normally undergo a lot of mutational events as they are developed. And this makes sense because a plasma cell's role is to produce a lot of immunoglobulins, a lot of antibodies, and we want to have a very wide repertoire, a whole variety of antibodies to all the different viruses and bacteria and other things that we're exposed to throughout our lives. But because they undergo these normal mutational events so often, they're actually more likely to have errors happen that then may trigger kind of a waterfall or a cascade of events that allows them to acquire more mutations over time and eventually become a cancer. And so that's in part why multiple myeloma and lymphomas are patient or are associated with kind of these mutational events, but we are getting better about predicting how they occur, how they occurred. But it can happen at a lot of different levels. And obviously this is a very complicated figure, but it can happen at the level of the DNA itself, the unwrapping of the DNA, or large chromosomal abnormalities, dysfunction in the packaging or shipping of the DNA itself. And so that's why we do focus a lot on the cytogenetics of multiple myeloma. And I'll go into that a little bit in detail as we go forward. So just to summarize what we talked about with mutations and how it relates to genetics, mutations can be point mutations where you have an alteration in a single letter or a single base, or they can be these large genetic events, what we call cytogenetic abnormalities. They can be inherited mutations that lead to specific genetic diseases or lead to these cancer predisposition syndromes where you would ultimately have to acquire another mutation at some point in your life in a different cell. Many of the cancers out there and many diseases are due to acquired mutations in individual cells throughout one's life. And those comprise the majority of cancer causing mutations. And as I mentioned, normal plasma cell development involves many of these normal genetic events to produce a variety or a robust immune system. But because of all these genetic events, it increases your likelihood of an error, which leads to a mutation that can lead to a malignancy. So let's hone in more on multiple myeloma and the genetics behind it. So the first thing I want to go through is what we're obtaining from the information we're obtaining from a bone marrow biopsy and the genetic information that can be used to inform patients and kind of an understanding of that for when you're having a conversation with your physician. So we look first at the number of cells, the number of myeloma cells in the bone marrow. We call that histopathology. We can take all of the chromosomes out of a myeloma cell and lay them out and look at large kind of genetic events. Normally you should have only 2 copies of the chromosomes, but here we can see in the first chromosome, there's an addition of something else. So that would be a genetic event that you might see in multiple myeloma. But really one of the areas we get the most information from is from this test that we do called FISH, or fluorescent in situ hybridization. So what we do with this test is we take the myeloma cells and we have these probes that are fluorescent. They light up under a specific type of microscope. And these probes attach to a panel of chromosomal abnormalities that we look for specifically. So you have to know the target that you're looking for. And so we have these panels of targets, and we can sit and we choose these specific panels because we know that they're involved in the disease generation, the pathogenesis, and we know that they have some implications from a risk standpoint. They can give us some prognostic information for patients. So I'd say of all the tests that we do in the bone marrow, this really gives us the most information to inform a patient as to what the risk might be from their particular myeloma. This is a really busy slide. I really include it more for reference that one might be able to go back to. And I apologize that it's small text and blurry, but the gist of what I want everyone to get is that these genetic events lead to, they can occur at different time points in the development of the multiple myeloma, and lead to the evolution of the disease from what we call a monoclonal gammopathy of undetermined significance. So where you have the detection of the monoclonal protein in your blood, but you don't have that many myeloma cells and you don't have any symptoms of myeloma. But as we acquire some new genetic and cytogenetic abnormalities, can progress to different types of myeloma. So from the FISH information, we can get information on the different types of genetic events, the cytogenetic events, and then we can provide information on the risk, the prognosis of their particular disease. How likely is it to come back after certain treatments? Should we tailor our therapy in a different way because of the risk? And I think that, like I said, we get some of the most information from these cytogenetics. And we know from clinical study that just in and of themselves, these cytogenetic abnormalities can give us information on the risk of the disease. And then we can also plug them into risk scores where we include not only the cytogenetic risk, but some of the clinical aspects of the disease itself, some lab abnormalities, and give a prognostic score to a patient and tell them this is what we expect the aggressiveness of the myeloma to be, and this is how we think we should potentially tailor the treatment going forward. Like I said, this is a busy slide with a lot of information on it, and I really just wanted to kind of highlight those key points. So in addition to looking at the cytogenetic abnormalities, which we do on a routine clinical basis, we can also look at the DNA string or the sequencing of the DNA in the myeloma genome. And we have done that. The first time it was done was in 2011, and it's been done a number of subsequent times as well. And we did identify a number of common driving mutations, but we realized that the myeloma genome is a little bit more complex than other malignancies, especially other blood cancers. For example, AML, there are not that many mutations that occur, and a lot of them occur at a relatively high frequency. So that allows for kind of more targeted sequencing in patients and also targeted treatments. When you compare that to multiple myeloma, there was a lot more complexity, some more mutations, and a lot of these mutations didn't necessarily occur that frequently, making it a little bit harder to describe how that impacts a patient's disease and helps with determining treatment. So we're a little bit behind other cancers, and we definitely have a lot of work to do in adopting gene sequencing in how we treat patients. And so there are a lot of ongoing studies to see how we can incorporate that into the clinical management of patients. One aspect we do use next generation sequencing for is for what's called minimal residual disease detection. And this is because myeloma cells have a unique genetic signature. As I said, they undergo all of these mutational events to create these antibodies, these immunoglobulins, and those mutational events become their signature that you can follow at different time points through the journey with treatment with multiple myeloma. And so we use next generation sequencing for that purpose, but we're still a little bit working on how we incorporate sequencing of the genome into actual targeting of the driving mutations. So one aspect that has been helpful is the participation of patients in some of these population-based and genomics-based studies. And so one success story is the COMPASS study, which was instituted by the Multiple Myeloma Research Foundation, in which they've enrolled over 1,000 patients across the across the globe and taken at least their baseline sample at diagnosis and sequenced them. So we've been able to characterize some of the mutations that are common and then associate those with different racial or ethnic backgrounds. And so it's because of the volunteers in these studies that we've been able to understand more. And this has become the springboard for that MyDrug clinical trial that I was talking about. And other genomic studies are likely to be the same as well. So just as an example of how this specific genomic study, the COMPASS study, has been used to highlight some differences in racial and ethnic groups, there was a study that was published a few years ago looking at the myeloma genome and comparing them between patients who self-identified as either African American or Black, or those who identified as Caucasian American or white. And there was a fair representation of both. Just from a population and demographics standpoint, one finding was that there were a higher proportion of self-identified African American patients who were younger at diagnosis. This group, comprising patients 40 to 49, was had a statistically higher group of younger of patients that were African American or Black, and that patients in the later age cohort were more likely to be Caucasian. Fortunately, from this study and several other studies ongoing, and that have been more recently reported, the survival and disease outcomes have been similar, and I think we've been better about achieving health equity. Of course, we still have a little bit of room to improve on that. And that maybe in African American and Black cohorts, the aggressiveness of the disease in general may be less aggressive, maybe a higher proportion of those lower risk cytogenetic abnormalities, although there's still a lot of work that needs to be done to really better define it. And one aspect of this particular COMPASS-based study is that we were able to look at the mutational profiles across these two groups separated by their ethnicity or race. And what we found was that there were a lot of similar mutations, especially some of the more common ones, but then there were certain mutations that were seen predominantly in African Americans and certain mutations that were seen predominantly in Caucasians. And what this allows us to do is take this information and then hopefully provide more tailored treatment options in the future, and gives us a lot more reason to be sequencing patients' genomes, their myeloma genomes, to provide that more personalized or precision aspect to their treatment. So as I said, we are kind of on the precipice of precision medicine for targeting these genetic mutations in multiple myeloma, and we still have work to do. But so far, we've been able to use a drug, venetoclax, for patients who have a specific cytogenetic abnormality. We're starting to target those BRAF mutations, such as the case presentation that I spoke about originally. I've talked a couple of times about this MyDrug trial, which is based on the genomics information that we've gotten from the COMPASS study and using sequencing of an individual's bone marrow to then find treatment options specific for them. But we still have a lot of work to do. We're still learning from the ongoing basic research, genomic research, population science, and clinical trials. And we're always hoping to continue to learn to getting to that ultimate goal of precision medicine and potentially cure for myeloma. And so it's really important that we include a representative group of all people in the research that we do, whether it's a clinical trial or whether it's this genomics or population-based research, so that we don't get caught in this one-size-fits-all paradigm. We can't assume that. We know the disease is developed differently, and so we're probably going to find that the treatments need to be different as well, that they need to be more tailored. So it's been a real privileged to present this to all of you. I've enjoyed it tremendously, and I have learned a lot myself. Obviously, if you have any questions, I am more than happy to answer them now. If I can't get you the answer now, I'm more than happy to reach out to you later about it. I'd like to once again thank the Myeloma Crowd for this opportunity, all of my colleagues who helped me design this presentation, patients and families and caregivers. Can't thank you enough.
Awesome. Thank you, Dr. Goldsmith, for that amazing presentation. We're going to take some time now for questions. If you are new to the Zoom platform, there's an icon at the bottom of the screen with different chat bubbles that has the Q&A on that. You can click on that icon and just enter your questions at that point. It looks like there's one question here. So Nell asks, given myeloma clonal evolution, how often should genome be sequenced? And she says, great talk. Hope the slides are available. Yes, they'll be available to you.
That's a great question that I don't have a great answer to at this point. So just to give a little background of what clonal evolution is. So as myeloma is treated, or as a patient goes through their myeloma journey, you can select based on the treatments that they receive for different clones or different offspring of the initial myeloma. It's a very complex subject, but some of those clones may be more likely to evolve or survive, whereas others may be more susceptible to the treatment that you're giving. And what we can see are these clonal tides as well. The clones that were predominant could then kind of wane, while others that are kind of at a low level can then take off. And so the question then becomes, how do you really know what to treat? I would say, anytime you're considering giving a targeted therapy when there's disease progression, genome sequencing as part of a clinical trial at this point would be really valuable when there's a shift or transition in the treatment of a patient. But we don't really use this yet, like I said, as a clinical tool. Hopefully we'll get to that point. Obviously there are a lot of barriers, the cost of doing it, the invasiveness of always having to do a bone marrow biopsy at disease progression. So I think the place where it really is the most relevant now is when you're discussing a clinical trial in which you're talking about using a targeted therapy. And then hopefully on that clinical trial, you might sequence it a subsequent time where you can look to see, did you target that clone of interest? Did other clones evolve out of it? So it's really a very relevant question that I think we'll hopefully have the answer to in the coming years, but I think it's going to continue to evolve just like the clones themselves.
Okay, great. Thank you.
So Carol wrote, how may one be tested for assessing which drugs may be of most benefit for a patient who has had the FISH completed? So that's an important question to really ask your physician as they're going through the treatment options. And unfortunately, we don't yet have so many targeted therapies for these FISH or cytogenetic abnormalities. The one I brought to light was this venetoclax for the 1114 translocation, which is one of the more common ones. We tend to use that particular treatment later on in somebody's myeloma journey because we're just starting to understand how effective it can be. But we do know at kind of a basic science or biochemical level that it makes sense. It's a target, it's rational. So for the cytogenetic abnormalities that we see on FISH, when there aren't too many targeted therapies out there yet, but we might, if we see a certain type of cytogenetic risk, might change the use or the length of use of some of the drugs that we have out now. So there's not a specific test for the cytogenetic or FISH abnormalities and which drugs we should use. It's more that we get that information back and then we say, this is the sequence or specific drugs that we might use. I hope that answered the question. I think, let's see, who was next? Do you know who was next, Valerie?
Yes, it looks like. And Dr. Goldsmith, you can see your slide so they can see you on the screen there. It looks like, oh, disappeared there. Lynette, maybe? This is joining late, but for those with a 1Q gain and
smoldering, IgA, kappa, 1500, bone marrow, 10 to 12%. Should treatment be considered at this time, or is there targeted treatment for this? So most targeted treatments at this point are going to be for patients with symptomatic myeloma who are going to be in later lines of therapy, just because that's how the drug development process, unfortunately, has to go. Hopefully, as we understand more about how these targets are used, then we can move them to earlier lines of treatment, assuming that they're also safe in addition to being effective. Trying not to give specific case advice on this, but I would say that for smoldering myeloma, we do have some patients who we would treat based on some other issues that aren't necessarily those specific criteria. So I think important to have a conversation with a myeloma specialist about, whether it warrants treatment for smoldering myeloma at a certain time point based on the data that we have available now. Okay, and then Eileen wrote, at the time of diagnosis, my genetics showed no mutations, deletions. Is it possible to have further testing done to obtain genetic information? How frequently does this occur? So if you're talking about the FISH testing for cytogenetics, I would say that any time that a bone marrow biopsy would be normally performed, that's when we would do it. So after a patient's undergone their initial treatment, after they've undergone a transplant, we usually repeat this and repeat the FISH testing as well to look for those large chromosomal abnormalities. And then oftentimes if the disease is progressing, it's not necessarily done as a routine monitoring because of the invasiveness and the kind of lack of utility of doing it repeatedly. But it is as far as like the sequencing of genes, that's predominantly done on clinical trials. And you can do certain gene sequencing panels, but I would say it's not routinely done at this point in clinical practice. Because in part, we don't yet know what to do with all of that information. And then, can you explain 1Q amplification, 17p deletion using the slides that were presented? Sure. So the 1Q amplification is going to be where you have more than one copy of the first chromosome, the Q portion, the long arm of the first chromosome. And that we are still understanding 1Q a little bit more. We do believe that it's a high risk cytogenetic feature and that patients may be more resistant to therapies because of that. But there are a couple of specific genes on that we believe, because of that amplification, because there are more of them, that is kind of driving their disease. And then 17p deletion is also one of the more high risk cytogenetic features. It contains TP53, which is a tumor suppressing gene. We call it kind of the guardian of the genome. And that that specific gene, when it's there, can lead to a whole cascade of DNA repair or terminate a cell when there's a lot of errors. So if that's missing, you don't have your guardian of the genome, then you're at higher risk of developing further mutations that can put things at risk of being resistant to treatment or becoming more aggressive. So that's kind of at a high level what those terms mean. For lambda 298 at present, kappa 0, what treatment more responsive? So looking specifically at the light chains, which is what you're referring to, the high light chain and the lambda and low kappa, we're not specifically treating based on whether you have lambda or kappa or the fact that this is more of a light chain myeloma, but we are using kind of the same drugs that we would use in the early presentation for that specific type of disease that we would use for most multiple myelomas. If there were other factors in that, as far as specific DNA abnormalities, or what I mean is more the FISH abnormalities, then we would, if there were more of those FISH abnormalities, then we might tailor the treatment a little bit differently up front, and then later on may incorporate some of the more targeted things depending on what we have. Lynette asked, can you explain deletion 14q? Is this high risk as 1q? So the 14q deletion is probably a little less common. Oftentimes we see 14 translocations, which is something where it is basically that part of the genome is put on, or part of that chromosome is put somewhere else and then leads it to be upregulated. 14q, I'm not sure of the risk of that specifically, and I think 1q is probably better described. Can you describe cyclin D1 in FISH analysis? So that's the 1114 translocation. It's cyclin D1 is involved in your cell cycle. And so it's one of those that controls the division and of cells. And so when you have that translocation, that 1114 translocation, it can make it so that there's some abnormalities in that specific protein that controls the cell cycle and allows the cell to escape the cell cycle and divide more rapidly. What abnormal genetics are unable to be detected after stem cell transplant in bone marrow biopsy FISH? So we do use, we do use the cytogenetic clearance as something as a little bit more reassuring that we've gotten more of the disease gone. It's probably not as sensitive as, or we know it's not as sensitive as minimal residual disease because it's still a small selection of the overall cells, but it is representative of, the cells a little bit. So it's helpful to know if they cleared if they don't have the cytogenetic abnormalities on subsequent FISH. It gives us, how we're more concerned if we still see the cytogenetic abnormalities after transplant. Then we're concerned that that recurrence is possible. But the MRD testing, as I talked about with the next gen sequencing, is more sensitive for picking up residual disease.
Awesome. Thank you guys. You guys had some amazing questions today, and this has been a great session. Before we leave, I did want to take a moment, turn the time over to Anna from the team. She's going to share with us how you can use what you've learned today to accelerate myeloma research. So Anna, the time is now yours.
Hello, Valerie. Thank you. Thank you,
Hi.
Dr. Goldsmith. That was amazing. My name is Anna. I'm part of the patient experience team here at HealthTree Cure Hub. And today I come here to invite you to our genetics study and real world outcomes. And what we're trying to find with this study is which treatments are providing the longest remissions and the best overall survival for the different genetic features. And as Dr. Goldsmith said, we won't find one cure for all myeloma patients, but what we need to find is the best treatment option for each individual patient. And that's one of the things that we're trying to accomplish with this study. Right now, we have 743 patients participating in the study. Our goal is 1,000, but hopefully we'll get more. And we're doing this study with Dr. Garrett Morgan of NYU Langone and Benjamin Diamond of the University of Miami. And if you want to participate, you can call us at HealthTree Cure Hub, and we can explain the process of how to do it. Our phone number is 800-709-1113. Again, that's 800-709-1113. Don't worry, that will be included in the email that you'll get after this presentation. And basically what you'll be doing is answering some questions. It's just 5 minutes, and sharing some of your medical information with us, like your FISH test, your treatments, and all the outcomes that you got from each treatment. This is an ongoing study, so we'll be analyzing data and we'll be sharing everything that we find out with you. And yeah, I think that's all. Thank you.
Awesome. Thank you, Anna. I'm looking forward to being a part of that study. So thanks for everybody attending. Our next month meeting is going to be held September the 23rd. We will have registered nurse Amy Pierre from the Nurse Leader Board to speak with us on how to effectively communicate with our medical team and medical professionals. Due to disparities within the medical field and the American culture of doctor knows best, it can be complicated and intimidating to speak to our medical team effectively. What should we do when we don't feel listened to? How can we know how to respectively communicate about difficult topics? When do we know when it's right to seek a second opinion or a new facility? As a Black nurse, Amy Pierre has been on both sides of this situation, and she wants to help us navigate the complexities of that. You may be also interested in other myeloma crowd community events that we have coming up. Friday at 1 p.m., our immunotherapy treatment chapter will have their meeting. Dr. Malin Holtkratz, I think, would join us to speak about a new therapy to add to the myeloma immunotherapy arsenal. It's called bispecific antibodies. And she'll be discussing who they're for, which patients are good candidate, what targets they're going after, what are the clinical trials studying, and what are the potential side effects of that. So definitely come and learn with us Friday. Wednesday, September the 1st, 7 p.m., the Muscles for Myeloma fitness chapter. It's going to be talking about the fitness challenge, the Move Spring Challenge that we're participating in honor of Blood Cancer Awareness Month. September is Blood Cancer Awareness Month. So this challenge is going to motivate us to move at least 20 minutes a day doing what we can. Learn more about this challenge and how to stay motivated by attending this event. On the second, we have two different events going on. At 1 p.m. Eastern Time, the stem cell transplant chapter will be meeting, and they will have Michael Riato. It's going to be talking about what happens with when a stem cell transplant doesn't go as we expected them to. Michael got an infection and other severe side effects during his transplant. And although it's rare, we want patients to be aware of what procedures like this, what things can happen during stem cell transplant, and offer you a greater peace of mind. And the same day at 7 p.m. Eastern Time, the MGUS and the Smoldering Myeloma chapter will be meeting. Dr. Jens Hillengrass will be sharing why he's studying the relationship between fitness and myeloma progression. It's definitely an exciting area of new research. On Tuesday, the 7th, the Myeloma Financial Coach chapter will be meeting again. We'll hear from Diana Valentine about good and bad debt and what it means for us as patients and caregivers. Thursday, the 9th of September, the Myeloma Caregiver chapter will be meeting. Licensed social worker Honora Miller
I think we lost Valerie. I'm glad her internet made it this far, but we'll just finish up by saying that we're grateful to our community event sponsors. We are really grateful to Dr. Goldsmith, and we're grateful to each of you for joining this Myeloma Crowd Community Program. And we hope to see you next month. So thank you for coming, and thank you again, Dr. Goldsmith. Take care.
My pleasure. Thank you.